We are starting 6.7 into this video, which is translation. We have already seen into the previous video that what was the structure of the tRNA. It look like inverted L if it is the 3D structure. It has got different stem and loop structure that we don't have to bother about. The two things that we have to see, one is the anticodon loop, that this is its 2D structure, as you can see right onto the left, and this is the 3D structure onto the right. It has got two main points that we have to pay attention. One was the anticodon loop, which is going to pair with the codon loop of the RNA. This is the acceptor stem onto which a specific amino acid, which is very much specific to the tRNA, is being attached. And same as here, that this is the site into the 2D structure, as you can see, and this is the codon which will bind to the anticodon loop, which is present right here onto the tRNA. The very first thing that is required in a translation process is activated tRNA, a tRNA which is bound with its respective amino acid. So let us say there are so many tRNA molecules that are just roaming around into the cytosol, and along with those tRNA we have those some amino acids. So we have already discussed that there are 20 kinds of tRNA into the cell, and we have seen why, because we have got 20 kinds of amino acid. So these are 20 kinds of amino acid, and which are going to bind to their respective tRNA. Now how this reaction is going to take place, we have got one enzyme which is known as the amino acyl tRNA synthetase. This particular enzyme actually helps this reaction to carry out. It takes the one particular tRNA and it helps, because it helps those amino acid and tRNA to get along with each other. And this, since this bond is since the reaction is the amino acylation of the tRNA, or you can say the charging of the tRNA. So as we can see right here, the process commonly called as the charging of the tRNA or the amino acylation of the tRNA. Since we are dealing with the ribosome as the place onto which the peptide synthesis is going to take place, let us take a brief look onto the ribosome. The prokaryotes have 70S ribosomes and the eukaryotes have the 80S ribosome that you already know from your 10th standard. But right now, one thing that several things that we have to keep in our mind. Number one, that what does this S stands for? This S stands for Swedberg. Swedberg is nothing but the name of the scientist who actually gave and made it a unit of the sedimentation. So if you're going to take a test tube, and into the test tube that you have into the solution, you have different kinds of ribosomes. So the ribosomes which belongs to the prokaryote actually are going to onto the upper side, because they are lighter as compared to the 80S ribosomes which are from the eukaryotes. And this will be the 80S and this will be 70S. Now definitely 70S ribosomes are composed of 50S and 30S, although 50 plus 30 is going to give you 80, but it is 70. Why? Because it's not simply about the size, it is also vary with the shape. So that is why that the sedimentation can vary. It is just not simply additive. So that's why we get two kinds of subunit, which is one will be the 50S larger subunit, 30S is the lighter subunit, smaller subunit, vice versa. And the same thing is true for the eukaryotic ribosome as well. The main thing that you have to keep in your mind is this, that they are composed of protein as well as rRNA. There are several types of rRNA into our cells, 5S, 20S, 23S, 16, and different kinds of rRNA. The main point is the 23S RNA and the 20S RNA into the eukaryotes, they both are acting as the enzyme. So we have already discussed that initially that there was one theory that all enzymes are protein, but no, indeed all enzymes are not protein. There are some exceptions, and this is one of those exceptions, that 23 and 28 sRNA are acting as the ribosome. They are able to catalyze the reaction, so they are ribosome. Now let us get back on to the NCERT. Let us try to see that it is also saying the ribosomes consist of structural RNA and the protein, and about 80 different proteins. Okay, in the inactive state, it exists in two subunits. So one was the larger subunit and one was the smaller subunit, as it is written right here as well. When the smaller subunit encounters an mRNA, the process of translation of mRNA to protein, it begins. The other thing, the ribosome, okay, so the ribosome also act as the catalyst 23S RNA that we just saw into the bacteria, and the 28 sRNA, 28S rRNA into the eukaryotes as the enzyme which is known as the ribosome. It actually helps to formation of the peptide bond. We have already seen that the very first codon will always be AUG, which also codes for the methionine as well. Apart from the starting codon, we have got some flanking regions that are present into the RNA. So if I consider this to be my mRNA, then I'll have my 5' site somewhere around here and 3' site, let us say here, and I've got some flanking regions onto the both sides. So I have this flanking region onto the 5' side and this flanking region onto the 3' side. These flanking regions are not translated, so they are untranslated regions of the cell of the mRNA. And somewhere around here, the very first AUG starts. Now let us take a brief look onto the mechanism of the process itself. So what is happening, that onto the ribosomes that we have got three sites. Site A, as you can see, these are three sites: site A, site B, and site E. E stands for exit site, A stands for amino acylated tRNA site, and P stands for the peptide site. So when what is happening, that once that we can see that peptide site is getting elongated, so what is going to happen, that this pink tRNA will be shifted onto the exit site, and this yellow tRNA, which with its respective amino acid, is going to add into the existing peptide chain. So as you can see that it is going to add right here, and it is going to come onto the P side. So from A side, it is going to come onto the P side, and the pink tRNA, which you can see that it can come onto the E side, and it can leave now from the ribosome. Although this is the 2D structure, if you like to get a glimpse that how does it look like onto 3D state, this is how it looks like. So you can see right here that we have got the larger subunit and smaller subunit. This is 30S, this is 50S, then it becomes the 70S. Okay, so this is for the prokaryotes. Here you can see that from one hole the mRNA is going inside, and from other hole it is coming out. This is A side, B side, and onto the back side, if you can see, the third tRNA is also there, which is the E exit site.